So hi, hello and welcome again, and today's topic, animal cells, an important and a large topic. And what I'm going to do today is we're going to look at the parts of an animal cell and the functions. And I'm also going to put some of my cheek cells under the microscope because after all, human beings are animals. We do belong to the kingdom of animals. And I'm going to show you how you can make animal cells visible under the microscope. We're going to start off first by making a drawing of an animal cell and by labeling it. And for this purpose, I have to clear my desk again, and I have to remove all of those parts that we will need later. So this in blue here, these are the functions of the different parts of a cell, and over here, these are the different cell parts. We're going to talk about the so-called cell organelles. These are structures that we find inside a cell. And here, a little sheet of paper where I'm going to make my drawing. So, I'm going to move this out of the way because I need a lot of space. When you make drawings on a sheet of paper, always use pencil. I cannot use a pencil right now because otherwise you're not going to be able to see it very well in the video. But you want to use a pencil because you have to erase sometimes some of the structures. I cannot do that, so it's a little bit risky. Do not draw the cells too small. It's very important, and leave enough space at the same time for labeling. You're probably going to write the labels of the cell by hand, and I will simply add those little pieces of paper here. So I always start off as a cell by drawing a so-called cell membrane, and this cell membrane defines the outside size of the cell. So what I'm going to do now is I'm going to make some labeling arrows later on, and then I'm going to add those pieces of paper here, the labels, but I'm not going to do this right away because otherwise I'm not able to draw properly. So this one here is the cell membrane, also known as the plasma membrane, and the biggest cell organelle, the biggest structure inside an animal cell, that is the nucleus. And I'm going to draw it as a circle. Many people, when they first draw a cell, they draw it far too small. They draw a tiny circle, but actually, as you're going to see very soon under the microscope, the nucleus is actually fairly large. And inside the nucleus, of course, we have the DNA. We have here the liquid part is known as the cytoplasm, and in the cytoplasm, what we have is those tiny little dots here. These are ribosomes, and these are structures that make proteins. Proteins are those things inside a cell that actually do the work. So the proteins perform a variety of different functions, many different tasks, and those proteins have to be made somewhere. And the ribosomes are those structures that make them. If you do not remember or if you don't know how to write those things, don't worry, I will be adding those things here as well. I'm going to simply place them to the side here because maybe I'm going to forget something. I don't want that. The mitochondria here. So I'm going to draw those as well here. The mitochondria, they are smaller than the nucleus. I draw it properly, okay? And what they have is they have infoldings of the membrane. This is not a very nice drawing here. But with a pencil, it's easier. And the mitochondria are responsible for providing energy to the cell. We also have so-called vacuoles. Here they are. Those vacuoles, they are able to store water and food. So we have vacuoles over here. These are like bubbles. Okay. And inside those vacuoles, you might have food. And you see, we're actually already finished here, and we can already start labeling. However, of course, there are many more different structures inside a cell. For example, in here, I should actually also include this here. In here, you have the DNA. You see, I don't even have a label here, so I'm going to add it later on by simply drawing a line. So, but now let's start labeling this here. I'm going to change the color, because it makes it easier this way to distinguish a little bit the different parts. How many of you remember what this is here? This is the cell membrane or the plasma membrane. Here it is. We put it over here, the cell membrane. Those dots here, these are the ribosomes. So actually I've only made the label to one of those structures here. Actually, there are many of them. So strictly speaking, I have to draw 2 lines because it's written plural. Mitochondria, some people call them the powerhouse of the cell. Okay, we'll always make sure that the structure that you want to label actually touches the object. The cytoplasm is not a cell organelle, it is simply the liquid content or the living content of the cell. So it's this, the fluid that fills the cell. The nucleus contains the genetic material and the vacuoles over here. Again, I have to have 2 lines here, okay? So, and of course, I also have to label the DNA, and I just see that you're not able to read this over here, so I have to move the camera across. So here we go, okay? That's better. Let's move the vacuoles over here like this. You get the idea, okay? And in here, I should actually also label DNA. So I'm going to write this over here. The DNA is the genetic material, can be found inside the nucleus. Okay, DNA. It's also a little bit sloppy over here. So these are the most important parts of an animal cell. As I already mentioned, there are more parts in there. I mean, you might have already heard of the RER, the SER, the Golgi body, and so on. I'm going to not mention them right now for the sake of keeping it simple, because what we're going to do later in a different video is we're going to compare the structure of an animal cell to a plant cell, and so there will be a few more structures here. So all of these structures that you see in here, or most of them, are known as cell organelles. Okay, the cytoplasm is not a cell organelle because it's not a structure, and the DNA is also not a cell organelle. I mean, it is something in here, but it is not surrounded by a membrane, because all of those structures here, like the nucleus, the mitochondria, the vacuoles, they are surrounded by a membrane, and therefore we call them cell organelles. So it's whether ribosomes should be considered cell organelles or not is a little bit disputed. Some people say yes, some people say no. But in any case, the general definition of a cell organelle is some kind of a structure inside a cell. So these are the most important parts of an animal cell, and pretty much all animal cells have those parts here. Sometimes there is not only one mitochondrion, but actually several of them. You know what? I'm going to draw a second one here as well, because I don't like the drawing here so much. So I'm going to draw another one over here. So there is a membrane as well, and there's even a second membrane which is too difficult for me to draw. And there are these infoldings. Of the second membrane to increase the surface area. Okay. So these are 2 mitochondria in here. They provide energy, they do cell respiration, and they're very important. So look what I've got here. You know this already. Controls what goes in and out of the cell. What is that? What is able to control what goes in and out of the cell? Well, it's the cell membrane, so we place it over here. Okay. Then the next one. It makes proteins. Look, the label is, I think, quite large, and I think there are even 2 of them in here. No, is it 1 or is it 2? Okay, it's 1. But it makes proteins. These are the ribosomes. They go over here next to the ribosomes. Should have made it a little bit smaller, maybe. The living contents of a cell, that is the cytoplasm. It's the liquid contents, or also the living contents of a cell. That is the cytoplasm. Prevents the cell from bursting. That's an interesting one. Well, that is a structure that is missing here. This is actually specific for plant cells. They have a cell wall, which animal cells do not have. So I'm not able to add this label over here. I have to keep it for later for the plant cells. It provides energy to the cell. It's this one over here. It's the powerhouse of the cell. These are the mitochondria. Again, I have to be a little bit careful here, because otherwise you're not able to read it. Okay. It stores food and or water. These are the vacuoles. And it contains the genetic material, the DNA, that is the nucleus. Okay. So I think it's probably not a good idea to add those descriptions right next to the drawing. You can see there is not enough space here. It does not look very nice. So what I recommend is that you label the drawing first, And then you make a separate table where you again write all of those parts of the cell, and then next to it you can write the definition or what it does. Okay, yeah, even this one, see, you cannot read it properly because my head was covering it. Okay, yeah, so yeah, it looks a little bit messy, I have to tell you. Yeah, so but at the end of the video I'm going to post again a picture where I'm going to rearrange everything more like in the form of a table, so it's easier for you to read it. Yes, what am I going to do now? Yeah, the thing that you've been waiting for. I want to show you now my cheek cells, and I'm going to do a little staining as well. So that I'm going to color it blue. It's relatively easy to do, but yeah, you need to have some ink as well, which I'm going to take from my fountain pen. Yeah, and you're going to see that under the microscope, you are not able to see all of those structures because many of those structures are simply too small to be seen by a light microscope. For example, ribosomes cannot be seen, but the nucleus, that is one of the things that you can see quite easily. Okay, so what I'm going to do now is I'm going to remove everything here, and then let's shift everything together. Okay, and let's start with the lab part. Okay, so. And I'm going to show you what you need. You need, yeah, here is a little bit of water because I'll be diluting the ink. You need a microscope slide and a cover glass. So this here, this thin glass is the cover glass. Okay, it's the microscope slide, cover glass. We need a little bit of water, tap water. Yeah, this, yeah, some cotton swabs, Q-tips. Yeah, normally you want to use them to clean out your ear, but I will be using it to now scratch off some of the cells from inside of my mouth. A pipette is always a useful tool, and very important, some ink. Okay, and what I will do now is I will start by preparing the ink because I want to stain the cells, but the ink inside of a fountain pen is too dark, it's too blue. So I have to dilute it a little bit. And I'm going to simply unscrew the pen, and look, there is an ink cartridge in here. So what I'll be doing is the following. I'll put this away here, and I will simply add a few drops of ink, 1 and 2 maybe, I hope, or 3. Let's add a third one. 3 of them in here. Let's put this back again, and we have to mix it a little bit. Just a second. Let me close this first. Okay. Let's mix this a little bit this way. Okay, I think yeah, it's still very blue, but not quite as dark as it was before. So, and now the fun part starts. Look, I'm going to take yeah, some fresh yeah, cotton swabs, and I'm going to now start scratching off the cells from inside of my cheek, inside of my mouth, and I'll be rotating the Q-tip to collect some cells. It's actually quite easy to do. It works like this. Mm-hmm. The inside cells of your mouth, they come off quite easily. Okay, they're always renewed and they're shed off. And now there are, I don't know, a lot of cells now on here. And what you do next, let's put this away, is the following. You simply take the cotton swab, and you, while rotating it, you simply, yeah, transfer the cells on here, and by streaking back and forth, you're breaking apart some cell clusters as well. Okay, and when you actually, I have to throw this away. I'm going to throw this away now. Okay, trash can. And when you hold it against the light a little bit, okay, you're going to see that there is some stuff on there, some tiny little dots, and these are the cells. And what we're going to do now is the following. We're going to take some of this ink, a small drop of the ink. We're going to put it directly on the cells like this, and the cover glass goes on top. And there will be a very thin film of ink now. The rest I put back. Okay. If you add too much of this, if the cover glass is floating, then you need some. Oh, I need to reach over to get some tissue paper. Maybe it's better this way. Okay, now you can see it much better. Look, there is a little bit of, so I'm going to simply remove the excess a little bit here. Okay, and you know what? Let's give it a try. Okay, so I'm going to switch over now to the microscope view, Yeah, and I'm going to put the slide now into the microscope. So here we go. You can actually see it now. Yeah, and up. Here we go. And I can already see some dots here. So let's see, I have to focus. Yeah, see, it works quite nicely. Those blue dots that you see, okay, these are the cells. Now, they're still quite tiny. So I'm going up with the magnification. So this is the 4 times magnification. Now let's go up a little bit. Now this is now the 10 times magnification. I'm going to now focus again. Yep. And it looks quite nice. You think probably, yeah, these are supposed to be the cells. Yeah, these are cells, and we still have to go up a little bit with the magnification. Let's look at the cell here in the center right now. So let's go up further. It's now 20 times. Here we go. Very nice. Many cells here. Let's look at the cell that's the other side. The right next to my eyeglasses. Okay, look at that cell. Maybe you see this. There's a dark dot in there. Okay, that dark dot that you see in there, that is the nucleus. Okay, you know what? I'm going to keep on readjusting this a little bit. Maybe it's better like this. A little bit more light. And I'm going to go up with the magnification yet more. Okay, now it's a little bit too close to my head. So let's move it over here. Okay. As a matter of fact, you see 2 cells, okay? The blue one on top and almost a transparent one right on the bottom. The transparent one, apparently, the ink didn't quite reach it yet, okay? But look at the cell on the top cell. You see that it has this dark oval dot. This dark oval dot, that is the nucleus. Yeah. You might also see the nucleus in the lower cell, which is kind of transparent and difficult to see. You see also a circle in there. That is also the nucleus. Yeah. So, but there are other cells over here as well. And look, if you look at all of those, all of them have this dark spot inside. Yeah. So that's always, each cell has this nucleus. Again, over here, there is a cell in the center, which is not blue. So maybe some cells did not accept the ink, or it might take a little bit more time. Yeah, again here, do you see a cell with a round dark blue circle? Yeah, here there's nothing over here. Again, this one over here. This one, these are nice. The one in the middle right now, you also see again this dark blue circle. That's again the nucleus. I think you get the idea, right? So those cheek cells, as you can see, they are pretty flat and thin. See, that's a nice one also here. Okay. Those cheek cells are quite, let me refocus, flat and thin. And a typical animal cells. You know what? Just for the fun of it, let's try it. If we can go yet 1 magnification higher. We're using this 60 times. Yeah, that's much better. Here we go. A little bit. Let's open the condenser a little bit. And some I bumped my fingers against the stage. So I have to be careful. It's that delicate, yeah. So I think, no, it's getting a little too dark maybe. Yeah. But I think I hope you get the idea, okay? So there's now quite a high magnification right now. You can also see that there are other things inside the cell as well. Huh? Yeah. The higher you go up with the magnification, the lower the light intensity is. And the more delicate everything is. Yeah. If you bump against it a little bit, then you immediately lose the focus. So you need a very calm hand. So yes, so these are my body cells, my cheek cells. Okay. The epithelium cells, because they're lining the inside of the mouth. Yeah. And they always come off, and new ones are always made. Yeah. So I think, here, see, that's a cluster. How many cells do you see? 1, 2, 3, 4, 5. Okay. And each one of them has this oval, round, now 6. Yeah. structure in it, that is the nucleus. Now you're going to ask, what about the other things like the ribosomes? They're too small. What about the mitochondria? They're too small. Okay, you need a different microscope for that, or different staining techniques. Yeah, those that specifically stain the different structures. Yeah. Yeah, so what I am going to do now is I'm going to simply call it quits for now. I hope that was that you kind of liked the video again. And in one of the next videos, we're going to have a look at plant cells. Okay, that's it for today. I wish you all the best, and see you around next time in the next video. Bye bye.